Hello, I'm Russell Burton from Forest Growers Research Limited, and I'm the Program Manager for the 21st Century Tissue Culture Program.
I am Jana Krajnakova. I work for Scion and I am the science leader for this program, which we are going to now talk about.
Yes, we look forward to taking you on an update of the Tissue Culture Program, which started 1 year ago. This program aims to make production of varietals of radiata pine more cost-effective and accessible to the New Zealand commercial forestry industry. The New Zealand commercial forestry industry has made large investments over very many years developing elite germplasm of radiata pine by means of different breeding strategies, with the aspiration to populate our commercial forests with those trees that create the best value for the industry and continue to grow that value. That is, those trees that enable us to continue to reduce rotation age, give us the best quality of wood within the trees, and maximise yield and productivity. And at the same time, and very importantly now, we need to ensure our trees are the most resilient they can be, as with climate change comes a heightened risk of their exposure to pests and pathogens. To fully deliver on growing the value of breeding, we'll need to transform our commercial forestry model from a family forestry model to a varietal forestry model. Varietals are trees with very similar DNA. By focusing on producing varietals of the best trees, we will increase the average qualities of important attributes such as biomass productivity, wood quality, and uniformity. Recent advances in tree biotechnology, including somatic embryogenesis, have enabled the development of a more flexible tree breeding, with genomic selection and deployment strategies over conventional seed orchard approaches. In addition, we expect to get deployment time down from typically 24 years to 6 years when genomic selection and somatic embryogenesis are combined. This slide shows the process of somatic embryogenesis. The pleasant protocols for somatic embryogenesis of radiata pine start with immature seeds, which are produced by crossing trees that exhibit the traits that we are after. The big advantage of somatic embryogenesis over other methods of vegetative propagation is that the embryogenic cell lines can be kept in a juvenile state by cryopreservation for later use. That means we can field test some of the genotypes and later focus on those genotypes whose performance can be assured, creating a further step change in raising the average properties of the trees in our forest. Our new program seeks to exploit the advantages of advanced somatic embryogenesis approaches and at the same time make a step change in improving the cost effectiveness of the production of varietals. We'll achieve this by using modern bioreactors and robotics rather than petri dishes. There's nothing new about creating varietals via somatic embryogenesis, with varietals produced this way already in commercial application, although in a very small way. Our business case for using varietals in commercial forestry tells us that if we could achieve about 40% of the commercial forest estate, we would realise at least an additional 1.5 billion per annum in value for the industry. What limits varietals increasing their share of new plantings is cost associated with small yields and the high level of manual involvement in the process. Typically, somatic embryogenesis has been based on cultivation on semi-solid media with several transfers required as the media needs to be adjusted based on the stage of development of the somatic embryogenesis. These barriers can be overcome by using bioreactors instead of petri dishes for growing the embryos, coupling this with automation and robotics, and as a consequence we can build a very high throughput propagation factory. This is the basis of this new tissue culture program, which is now 1 year old. Essentially, our plan is to move from the petri dish-based propagation to high throughput bioreactors and couple this with automated embryo sorting and germination. What our program will do will be to replace many of the traditional operations by using bioreactors, specifically a bioreactor called a temporary immersion bioreactor. They called this as the liquid which provides the growing stimulants, temporarily floods, then empties from the reactors. The environment inside this reactor can be very closely controlled and changed as required to induce the embryos to develop and produce hundreds, if not thousands, of plantlets. This creates better access to the key components in the nutritional media, higher proliferation rates and increased production of the somatic embryos. There are a lot less manual steps and provides scalability through the exploitation of robotics and automation. This approach offers several advantages over manual handling and using petri dishes. The use of liquid media also has an advantage in simplifying the manual handling. When the somatic embryos are ready, they can simply be transferred into another vessel, where the wonders of artificial intelligence and robotics can decide which are the good quality plantlets, remove those which are not properly developed, and then transfer the good plantlets to a soil plug, ready for the next stage of their journey to the nursery. Although we are pioneering this for radiata pine, aspects of the approach we have just described is already well advanced for some other species. We have forged some relationships with other organisations internationally, and as a consequence been able to leverage off their knowledge as we build a system that works for radiata pine. This last year, the program's first year, has seen us consolidate and formalise those relationships. And in spite of the challenges thrown at us by COVID-19, we have succeeded in building at Scion the first ever facility purpose-built to house the temporary immersion bioreactors focused on the tissue culture of radiata pine. Let's see it in operation.
I would like to take you now for the virtual tour to Scion laboratory dedicated to bioreactors. This laboratory has two independent rooms, bioreactor room and preparation room, where we have laminar flow for work in sterile conditions. Now we will be entering to the bioreactor rooms where we have control airflow. Then temporary immersion bioreactor setup systems work with compressed air, which is going to be explained later on. The bioreactors are connected with automated control with computer software called SoftRun. Now I will talk a little bit about technical design. Scion has compressed air running below the floor, and we have moved it up and compressed air is now in a steel pipe carrying to the air filter, and then we control also the pressure. In present moment, we work with the pressure 7 kilopascals. Then filtered air enters the solenoid valves, which could be digitally or manually controlled. Software opens and closes the valve according to user's instructions. When valve opens, filtered air enters the right tube. And this supplies a whole bank of bioreactors. The bioreactors are controlled also manually using manifold. Therefore, we can switch on or off bioreactors which are not in present use. Tubing are connected to bottles containing culture media. Compressed air passes another air filter unit. And then pressure of compressed air displaces media volume. Media flows upwards through another filter and it is reaching bioreactor. When the solenoid valve closes, media drains back to the bottle. Now we are going to demonstrate how we work with plant material. For education purposes, we have used blue dye. When material is in bioreactors, then also media is poured into the bottle. Everything has to be closed and sealed properly, then moved to bioreactor's shelf, and compressed air tubing is attached with bioreactor. The connection between bioreactors and the computer is done using special software, SoftRun. Here we demonstrate how we operate using Bank 2. We are going to set our software for 2 immersions, which are going to start at 9 in the morning and then at 9 in the evening. And the duration of immersion is going to be 25 seconds. It is time when the solenoid valves will be open. We are now demonstrating what is happening when the solenoid valve is open. That medium goes up, it is reaching the platform which is holding plant material, and it is bringing fresh medium. Plant tissue is nourished with the new medium for a period of 25 seconds. It is really important to control that the medium will not rinse the plant tissue into the medium. When the immersion cycle is over, as you can see, medium drains down into the bottle. And this cycle is about to be completed. Now, short demonstration how embryogenic material look like after 14 days being cultivated in bioreactors. And these are our current experiments.
As we indicated, we have a large number of partners and collaborators involved in the program. We're able to leverage off a lot of that knowledge by working with organisations such as the Georgia Institute of Technology, the Finnish Natural Resources Institute, and so many other organisations. Most especially also the New Zealand industry, which are providing a lot of input into the program design and development and bringing their experience within as well. Also acknowledge the support from the Forest Growers Levy Trust and the Ministry of Business, Innovation and Employment. So in conclusion, our program aims to add some significant new tools and knowledge to our industry by harnessing the power of bioreactors, automation and robotics in the production of varietals using somatic embryogenesis, making this accessible to all those in the industry by improving cost effectiveness and reducing deployment time. Another key step is building our knowledge with industry and adding value across the entire industry. We thank you for your time and we hope you found this presentation of interest. On behalf of Jana and her very extensive science team and our industry support team, I thank you for your time and interest.